Hey, good evening, everyone, and welcome to today's session. Today's session is all about the questions you have asked, and we are going to answer them one by one. Okay, so let's quickly start. Um, I will start with the bottommost question, which is the topmost actually. The question is how to collect and transport environmental sample for analysis. What are the types of environmental samples? See, what kind of analysis you need to tell us. For example, we can collect sediment and isolate bacteria and go for its species identification using DNA sequencing, or you can collect, air sample through filter paper, even unculturables. The bacteria that cannot be cultured or yet to be cultured can be sequenced from the filter paper particles that is being collected. So if it is a filter paper or something, better to preserve in the lysis buffer and transport to the company. Or if it is an environmental sample, you have to just talk with the people in the company and before you courier those samples. That can be accepted in under this program. And ClusterLex software is buffering. I'm not responding. Tried reinstalling also and keeps happening. See, you need to have enough working space in your computer, at least 10 GB, I will say. If the computer capacity is 240 GB, at least it should not be filled more than 230 GB. Okay, if you have a 10 GB, this non-response will not happen. Usually, these non-response are because of lack of memory in the system. So I would advise clear the temporary files, egg files, using your antivirus software or any special softwares. Clear them, have more space. Try again. ClusterLex is working fine for many. Okay. In this session, I have a doubt regarding why it is necessary to merge forward and reverse sequence. It is necessary because, like I mentioned during my introduction class, the current sequencing technology, sequencer, especially Sanger sequencer, they cannot read more than 900 to 1000 base pair at a stretch. Similarly, the tag polymerase you are using in the PCR cannot, amplify the gene length more than 700 or 800 base pair. That's why for 1500 base pair, you need to split the sample PCR amplicon precisely into two for single strand amplification. One is forward strand, another is reverse strand. Both are compiled in the after machine output file. Both are merged to extract the target length of your gene. If you are using either one of the strand, you may end up having only 60-70% of the length. Also, merging act as a complementation and as a quality control that whatever the sequencer has produced is actually a reliable result because the complementation can be easily checked and that's act as a quality for the sequencing reaction. Okay, that's why we are going for merging using bioedit. So I have few doubts to be clarified. When query sequence shows 100% similarity match for more than 3 species, meaning they are not a molecular marker or they cannot be a barcode gene for that particular species. If you have attended yesterday's multi-locus gene sequencing webinar, I have shown examples of Bacillus. This is bound to happen with Bacillus. That's why we are having alternative genes like RPOB and other genes. That's why under the program currently you guys are doing, you have a privilege of sequencing two sequences from one sample. Okay, one bacteria, two different genes, or one plant sample, two different genes, both RBCL and MATK. You can either choose. If that plant particular species have this problem of the DNA barcode not working precisely. When I say not working, they are not identifying the plant species belonging to single species, but as per your observation, they are matching with 3 species, so they cannot be a reliable barcode. Which is the best to compare between query coverage or species identity? Percentage of identity. Yes, percentage of identity is the first criteria you should choose. Second comes the query coverage. Okay, so when you have a percentage of identity 100, most probably it will be a same species. Okay. When we run the blast search using DNA sequences and get it 100% identity matches to more than one species, just now we have addressed here. So that could not be a same species. Okay, what are the that cannot be a appropriate barcode. What are the other bioinformatics tool available for reverse complement? Yeah, many tools. Even you can do reverse complement in Mega in your multiple sequence environment. I want you guys to realize these Mega, BioEdit, and ClusterLex, They can do multiple function. We are using for a specific objective doesn't mean that they will work only for that. Okay, I encourage you guys to explore other options. For example, may have a molecular clock, evolutionary time, nucleotide diversity can be calculated. All those sessions are lined up for us. We will be seeing them one by one. I'm just waiting for from your side. I'm just waiting for you guys to send the samples to us so that we can have our mission output files and other files ready. We can use our own sequences to learn other options in these softwares. So today it's all about addressing these questions. If you have more questions, please do use daily reflection form that is pinned in our group every day so that I can get back to you. And I'm still waiting for your microbial cultures and other samples. Okay, once the sample reaches the company, the same day within 3 hours, they will acknowledge you. You will know that it is reached. After sending the courier tracking number, please do track your courier. Okay, keep on tracking it. When it is delivered, you can be sure within 3 hours they will acknowledge you through WhatsApp. Okay, and those who have already sent the sample, sequencing is in full fledged. You may receive your results before this Sunday. So today there will be no live sessions. We thought we will talk about the commonly asked questions. I hope the analysis is coming fine. And before tonight, I will share the Google form to check your phylographs. Thanks for sharing. And thanks for joining us today. Thank you.